Hi, my name is Melissa Miller, and I work as a wildlife veterinarian and pathologist at the California Department of Fish and Wildlife Office of Spill Prevention and Response. I'm based just next to Monterey Bay in the center of California. The topic of my presentation today is interesting examples of pathogen spillover as taught by California sea otters. What inspired this presentation for you is our current pandemic, which is obviously affecting all of our lives, and has really brought to the forefront this idea of disease transmission between animals and people. The virus that we're talking about is a coronavirus called SARS-CoV-2, and the disease that it causes is called COVID-19. This particular virus is thought to have originated from some type of bat and may have reached humans either through some type of unknown, currently intermediate host, or may have found humans directly from bats. That's still being studied by a number of scientists and is currently unknown. So what we'd like to do is look back at California and investigate some of the examples of pathogens finding their way into unexpected places. But in order to do that, we first have to learn some basic vocabulary. The first of the terms we need to understand is, what do we mean by a pathogen? And really what that is any type of bacteria, parasite, virus, or other microbe that can invade animals, humans, plants, fungi, basically any type of organism, and cause disease. The next term is zoonosis. And what that is a disease that's caused by pathogens that can spread between animals and humans. And so as an example, zoonotic pathogens cause zoonosis, or disease due to this pathogen. Traditionally, we think of these as pathogens or infectious microbes that go from animals to people, but in truth, they could go either way and go from people to animals as well. And if you've been watching the news, you may have seen the current report of a tiger at the Bronx Zoo that tested positive for COVID-19. And one of the suspicions is that it may have acquired the infection from a human. That's still being studied. Another term that we want to understand is, what do we mean by pathogen spillover? So what that means is a pathogen from one species, and that would be a species that's very commonly infected at high levels with that pathogen, moves into or colonizes a new host and may or may not cause a disease outbreak. Under some circumstances, that pathogen then may be transmitted from individual to individual within the host population. And so the COVID-19 outbreak is a good example of this, where bats may have been the source. Through some route, it reached people, and now what we're seeing is transmission between people. So we've got spillover into people, and then spread directly between people, which can cause amplification of an outbreak and lead to epidemics or epizootics. So when we talk about this next term, pandemic, what we really mean is a disease outbreak that occurs over a wide geographic area and affects a high proportion of the population. And really what we're talking about there is an epidemic, an outbreak of disease that's happening on a global scale. And an epizootic is a similar term, but it just means that it's happening in animals. What's really interesting and important to note, though, is that pathogen spillover events are happening all the time. This is not a new concept. It's not something that's just happened now. In truth, these war games between hosts and pathogens have been going on for many, many millions of years. And pathogens like bacteria and viruses are highly adaptable and readily seize opportunities to move into new hosts and colonize new habitats. The good news is that most of these spillover events are limited regional outbreaks, and they don't get to the scale of the COVID outbreak that's happening right now. But it's important to note that human activity can lower barriers for spillover to occur, facilitate spread of these pathogens to larger areas, and because of these things, they can make events more severe, and that's exactly what's happened in the case of this COVID outbreak. Examples of this that have happened in the past include the HIV virus, Ebola, MERS, and both of the SARS viruses. So we're really going to learn more about this on a California-centric basis now, and it's really going to be taught to us by one of our most iconic mammals that lives along the central coast of California, the southern sea otter. And these very beautiful, unique animals live right along the shoreline. They consume about 25% of their body weight in prey each day, including all types of invertebrates. And what's interesting is that many of these prey items are really efficient at concentrating pathogens that are flowing through water. And because of this, it makes otters extremely sensitive as environmental sentinels. And if you pay attention, they can teach you a lot about what's happening in their environment, both on land and in the adjacent nearshore habitat. And as part of this today, we're going to learn about what's the connection between all these different species that at first it seems like, geez, maybe they're not really that tightly connected. But what you'll see is that through learning from sea otters, we've realized how closely connected we all are to each other. Everything from foxes and bobcats and mice and birds and people all have direct or indirect connections to sea otters. The other thing, too, is I want to kind of challenge your thinking about what the word habitat means. Traditionally, we think of habitat as the top two pictures of forested land or river valleys, and those provide critical habitat for mammals and birds and that kind of thing. But if you think about it from the perspective of the pathogens, their site is a little bit smaller, and they're looking at individual animals as a habitat to colonize, and from there use those animals to spread themselves around. And so that's an important concept to keep in mind, is that they're looking at the world from a little bit different perspective than we are. And when you take animals that are infected with these different pathogens and move them around, that can actually produce or facilitate pathogen spread. So now we're going to dive into our program, and you're going to join us to help investigate some mysterious sea otter deaths. What you see on the left is part of our crew getting ready to examine a sea otter that has been found dead, to try to figure out what happened to it and see if there's anything else that we can do to address that for the living sea otter population. And from there, we'll collect little bits of tissue and look at them on the microscope. And as you can see, we've saved a place at the microscope just for you to join us. So have a seat and let's go. So our first case is the case of a dead sea otter on the beach. When we did our postmortem examination of this otter, we really didn't find that much. Maybe some mildly swollen lymph nodes, but really not much else. So this is a very common finding, and it really emphasizes the importance of going beyond that and looking at tissues at that higher level on the microscope. When we did, what we found was this. This is a very high magnification view of tissue from the otter, and those little tiny pink and black dots on the left where the arrow is are single-celled parasites called protozoa. And the picture at the right is if we had an electron microscope and we could blow that up and take a bigger look, you'll see that the individual parasites are banana-shaped, and they've proliferated into a little package that then can break open, and each of those bananas can go out and infect another cell. So the appearance of this is very typical for a parasite called Toxoplasma gondii. It's a protozoan or single-celled parasites where cats of any type, so that's mountain lions, bobcats, domestic cats, are the only hosts that are capable of shedding an environmentally resistant egg or oocyst, and that egg can then in turn infect a wide range of animals, including humans, and if it makes it down to the ocean, sea otters. What's important to keep in mind about this is that this is an example of an introduced definitive host. We know that there were cats here along the landscape in California before people came along, but I think you'd agree with me that the number of cats has drastically increased as humans have colonized our coastline. And it's also important to recognize that there's no marine source that's known for Toxoplasma gondii. This is a land-based pathogen. So what do we know from our study so far? That on Toxoplasma, we know that cats are the source and the only source that we know of for sea otters. In spite of that, 62% or more of California sea otters appear to be infected with this parasite. And it is a relatively common cause of death, but it's not transmissible between infected otters, except that it can pass through the placenta and cause damage or death to the fetus. And we also know from our research that there are high and low risk areas along the coast for infection that may have to do with land use patterns or other things that we don't quite understand yet. The high risk areas are in red in this figure on the right, and the low risk areas are in blue. So our next case is the case of the sickly sea otters, where we had 40 sea otters that stranded over a small coastal area with very severe seizures and died within hours to days. When we did the postmortem examination, we really didn't see that much, maybe a little bit of swelling and edema around the eyes, as you can see here, and some tiny pinpoint hemorrhages that we found under the skin of affected animals, as you see on the right. But when we got to the microscope, and this is a high magnification view of the brain, what we found were the same thing, little single-celled parasites that had infected cells of the brain tissue and were proliferating, causing these flower shapes called rosettes. And that's a sign that the parasite is budding off in all directions and getting ready to break out of the cell and go and infect new cells, which is part of the reason that these can be very pathogenic and kill sea otters. So what we know from that type of morphology and subsequent testing is that this was a parasite called Sarcocystis neurona, and the life cycle is very similar to Toxoplasma gondii, except that the definitive or egg-shedding host is a possum, not a cat. And on infection, these possums can shed millions of these eggs, which can make their way to a wide range of hosts and cause nervous system disease, muscle damage, and a wide range of other types of problems. So this is, again, an example of an introduced definitive host. Possums aren't native to California, and there is no known marine source of this parasite. From our studies at this point, what we know about Sarcocystis neurona is that possums are the only source, but in spite of that, over 50% of California otters are infected with this parasite. It's a very common cause of otter death, and as you've seen, it can cause outbreaks of disease. It's not directly transmissible between infected possums. We're still trying to work out how common, or if it does cross the placenta. This is not something that affects people that we know of. But when you look across both parasites now, Toxoplasma and Sarcocystis, now we see that the only sea otter source for both parasites is terrestrial animals, mammals. And in spite of that, over 75% of tested California sea otters have one or both parasites. They're infected with those, which is pretty amazing when you think about it. And also, like Toxoplasma, we found that there were high-risk areas for otter deaths due to Sarcocystis along the coast, as shown here in red. Our third case is the case of the sea otters with creatures in their brain tissue. So this is a case where at the postmortem examination, we didn't really see much that informed us as to how the animal died, so we collected tissue to look at on the microscope, and much to our surprise, we saw these big things in brain tissue accompanied by a lot of inflammation, which is what all the little dots are around there. Those are white cells. And these creatures are very large, and it turns out what we're seeing here are sections through worms in the sea otter brain. So in investigating this, what we found is this is most likely Baylisascaris procyonis, the raccoon roundworm. So this is a similar type of event as to what we've already talked about, where raccoons are the only egg-shedding host. They pass these eggs in their feces. They're very tough, environmentally resistant, and they can make their way down to the ocean and affect sea otters, and also can affect people as well. So this would be an example of a human influence disease, where when we move into habitats and we leave out garbage or we provide cat food for our pets outdoors, it can cause raccoon populations to increase and cause this problem to worsen or make more risk for downstream animals such as sea otters. And again, it's another example of a disease where there's no known marine source of this parasite. It's definitely coming from land. And as we've seen so far, all three of these first examples are examples of terrestrial hosts that defecate environmentally resistant eggs, which flow to the ocean, can get into prey species or contact the animals in the environment and from there infect marine wildlife. So let's look at another example of a different way that pathogens can make their way into marine mammals. Case number 4 is the case of the rapid sea otter deaths with only moderate lymph node swelling found at necropsy. So obviously we had to go collect tissues and look on the microscope. And the picture on the right is an example of a lymph node looked at that's on the microscope at very high magnification. Most of the little dark dots that you see are the nuclei of white blood cells. And the arrows are pointing at little round reddish dots. And what we're seeing there are what are called inclusions. And those could be in the nucleus, in the cytoplasm of the white cells. And what those are an indication of is that this otter was infected with a virus. What type of virus? What do you guys think? Maybe you can guess. It turned out that the otters were infected with a virus called canine distemper virus, which is a morbillivirus. This virus has a wide host range, but especially you can see it commonly in dogs, foxes, coyotes, other types of canids. And interestingly, some sea otter deaths coincided with the CDV outbreak in gray foxes in the same area and during the same time period. And the picture on the right is where I took a swab and I collected some cells onto the swab from a fox that I thought might have died of distemper, and I just smeared it on a slide and stained it. And what you can see is a cell that's laid out on the right-hand side that has those same pink dots all through the cytoplasm, indicating that the animal was infected with canine distemper virus. So our last case, we're going to talk about bringing it back to people now, and influenza viruses or flu viruses. And you may have remembered that in 2009 there was a global human pandemic of the H1N1 influenza virus, which was also called swine flu. And some of the earliest cases in the United States were actually recognized in people in California in 2009, March, April of 2009, as you can see in this published report on the right. The source of the H1N1 virus was thought to be mixing viral genes between birds, pigs, and humans, but this pandemic, as you'll see, caused very severe effects globally, including in the U.S. So this picture shows you just numbers for the first year of the outbreak in the United States, from April 2009 to April 2010, when over 61 million cases were recognized, and there were at least 12,470 human deaths due to this virus. So what we were curious about, ourselves and other scientists, were could we find evidence that this easily transmissible virus could have also reached marine mammals as well. And so we tested sea otters here in California for antibodies to the H1N1 flu virus, and other scientists tested a wide range of other marine mammals too, and the results are absolutely fascinating. In California, sea otters, sea lions, harbor seals, and elephant seals were found to have antibodies to the H1N1 virus. In Washington, sea otters too had antibodies. And on the east coast of Canada and the U.S., gray seals were found to have antibodies to the same virus. And interestingly, all of these different groups of animals developed these antibodies for the first time that was detectable in either 2009 or 2010 onward, so after the peak of the event in humans in the same area. And for these last two pathogens that we just talked about, the CDV and the flu, as well as the current pandemic of the SARS-CoV-2, the classical route of infection is through respiratory droplets that are expelled through coughing, sneezing, and that kind of thing, and can make their way into other humans or potentially into animals as well. And so in the case of the marine mammals, we've seen antibody evidence that these animals were infected, less evidence that it was causing major mortality in those animals. We didn't find any for sure cases in sea otters at all in our work, and mostly these different studies really found the presence of antibodies showing exposure, but not really documenting severe disease in the marine mammal populations. So to summarize, I hope you've seen that humans and animals are capable of sharing many pathogens, that pathogen spillover events are common, but few develop into pandemics, that human activities can enhance the risk of pathogen spillover and spread through things like exploiting wildlife resources, intensive agriculture, habitat destruction, and animal translocation. But it's also important to keep in mind that we can help prevent pandemics through our actions as well. And so bringing it back to Earth Day, I hope that from this you have learned that wild animals can teach us a lot if we pay attention. They've shown us that we all share one small planet. Our actions can negatively impact animals and habitat, but again, those impacts can then come back to harm us, human populations. And I hope that you've learned from our work that this type of research about learning from past events is really important, and that our continuing conservation efforts will help to keep things in balance and provide direct benefits for people, habitats, and wildlife. And I hope that you try your hardest to celebrate Earth Day this week, but also strive really hard to celebrate Earth Day every day. Do what you can to protect our planet, its beautiful wild things, and its habitat, and help protect the people around you through your actions. If you want to learn more about this, here's a number of sites where you can learn more about oil spill response involving protection of marine mammals, marine mammal anatomy, pathology, and diseases. So check those out if you're interested. And on behalf of Elsie, Minnie, and I want to thank you very much for your attention, and we hope you enjoyed our presentation. Thank you.